So good morning, everyone. I'm really delighted to be able to chair this session this morning. I'm Seychelle Voss. I'm a faculty member in the biology department at MIT. And our first speaker this morning is Dariusz Plewinski from the University of Warsaw. And he will be telling us about the spatial chromatin architecture alteration by structural variations in human genomes.
Hi, hello. First of all, I would like to thank organizers for inviting me to give a short talk about our recent results, which are related with the fundamental question, which is namely how the spatial chromatin architecture is altered by the structural variation in the human genomes. So how the changes in sequence of DNA sequence of which can be observed in a human population is translated into differences in the three-dimensional structure. So that's basically our idea of our work and the recent results. I am co-affiliated at Warsaw University, which is a center of new technologies, and at the same time I'm also co-affiliated at the Warsaw University of Technology. And that's which was interesting for us, and it was somehow founded on the fundament, on the basis of the 4D nucleome project, which was initiated by NIH, and but also not only this project, but also ENCODE start to provide three-dimensional NGS-based data to the scientific community, and those consortia allows actually us to understand what is the variability of the 3D structure of chromatin across different cell types. Okay, so that's important because then this knowledge can be extended based on the sequence information we have, which is collected for multiple individuals in the human population, how these structures would be affected by the sequence variability within the human population. So the concept, so our participation in 4D nucleome was focused on a specific approach, which is ChIA-PET, and I will talk about it shortly, and also about imaging studies, which allows us to build the effective, very high throughput modeling platform, which allows to construct the 3D structures, 3D models of chromatin 10 nanometer fiber, and based on a sequence information and a reference 3D structure. Okay, but coming back to the original question, so we have sequence, we have structure, and we have function. Okay, and the problem of relation between sequence, structure, and function in the case of chromatin is definitely a multi-scale problem, and it reflects the complexity of human genome. Yeah? So we start with double helix with the size 2 nanometer. Then we have nucleosomes which DNA is interwoven around, and it provides so-called 10 nanometer chromatin fiber, and then the whole structure of the in this case human genome is being constructed. So we have very complex, very long and very complex biomolecule, which we are trying to analyze. And this multi-scale organization is also reflected in the context of function. So we have certain regions which are critical for specific genes or specific enhancers. So for example, chromatin looping allows you actually to see interactions between promoter and enhancer regions. On the other hand, there are so-called topologically associated neighborhoods in which certain sets of genes or enhancers are working together in a more or less coherent way to provide a specific biological function. And so on. So there is chromosomal territories, there is A and B compartment, meaning heterochromatin, unichromatin. There is also an important biological role of nuclear lamina, which silences some specific regions of DNA. In terms of modeling, actually, the problem is extremely complex. And if you start with modeling, like by quantum mechanics or by molecular dynamics single atoms, then it will be very challenging in terms of pure raw computing power to model something larger than the small actual fragments of the whole 2-meter long chromatin, which we observe in a human genome. Okay? So the microscale itself, multi-scale, and also macroscale, again, is also have different scales, spatial and Tourette scales, which can be observed in the genome. Okay, so what are the two fundamental methods which allows actually to identify experimentally some structural information about the chromatin? So two general technologies are either based on a microscopy. In this case, I'm showing 3D fish, in which you can observe like two different genome clochi very precisely and, for example, calculate the physical distance between them. So the physical distances would be R here, and S is the distance along the chromatin fiber. Okay, so you have to like go through the whole fiber and calculate what's the distance between along the fiber. And then you can try to correlate based on observing multiple loci, pairs of loci like that, what will be the relation between linear distance and physical distance. In the case of MGS-based methodologies, what people observe is not only a single pair of two genome clochi, but actually all against all. But what you're observing is not precisely the physical distance like in the microscopy, but rather a certain probability of a contact across different cells. So to get such a probability of contact map, which is, as I said, all against all regions in the given segment of DNA, or maybe it could be the whole chromosome or even the whole genome, and then you can observe which regions tends to interact to be close to each other as compared to other regions which tends to be actually far away from each other. Okay, so what we learn up to now from microscopy, first of all, this is the fundamental observation that chromosomes does not mix, do not mix. Okay, so they occupy separate subvolumes of the nuclear space, and therefore, if you use some oligopainting to color a specific chromosome, then those colors will not mix. So they will actually stay differently. So that it means that actually it's not a random polymer. It's actually a very well organized cellular nuclear machinery. Moreover, if you go to higher resolution using, for example, electron microscopy or some super resolution light microscopy, you can observe that generally speaking, nuclear volumes divided into dense regions and sparse regions. Okay, and the sparse regions has a lower density of chromatin, whereas the dense regions they are characterizing inactive regions, inactive nuclear compartment, heterochromatin, which is actually harbor genes which are kind of switch off. So you can approximate the nuclear structure of the nuclear volume with the swamp-like landscape in which you have channels which allows you to transport quickly some biomolecules, like the channels here, and also some islands of dense regions which definitely slow down the interaction, slow down the transport, and so on. Going back to the genomic methods, two methods are actually fundamental, and if they were introduced in 2009, fundamental to understanding the all against all interactions for in this case human genome. So high C and Chirapet. In the case of high C, you get precisely the heat map which reflects the probability of interaction across different all regions, all segments of DNA. Here we have like chromosome 14, and of course it could be another chromosome or whole genome. the case of Chiapet, there is additional step, which is related with the immunoprecipitation. So this step selects from all those interactions which are observed in high C, very specific interactions which are related with the protein mediator. In this case, that could be in our approach. Actually, we are using data mostly from two proteins, CTCF and RNA-POL2. So those arcs, so therefore we don't use heat maps like here, but we use arcs, and each arc represents a single interaction between beginning and end of the such so-called chromatin loop. And this beginning and end is glued with the given protein, so the CTCF in this example, or it could be RNA-POL2. So there are similarities and differences between those methods. Chiapet provides high resolution without deep sequencing, so this is cost-effective. On the other hand, Chiapet is very challenging experimental technique, so it's not easier to perform such an experiment. High-C is much more easier to be done, but you have to sequence deeply to actually start to observe the chromatin loops. Okay, so the understanding of genome, which is rooted in the observations, either High-C or Chiapet, is that we indeed observe two phases in the genome, so dense and the sparse phase. So the dense would be heterochromatin, sparse with euchromatin, and they tend to form separate compartments, and those compartments therefore reflect the differences in the function performed by these regions, which are either in heterochromatin or euchromatin. But moreover, if you go to the higher resolution, you start to see genomic domains, so certain globular structures along the chromatin fiber, which form so-called paths or topologically associated domains, which allows actually builds like a local neighborhoods which harbors genes or enhancers. Okay, so the multi-scale nature, multi-scale organization of chromatin is reflected here. We start with whole genome, then the whole chromosome compartments, domains, and finally chromatin loops. And it was already proved that changes at the level of sequence, like deletion, for example, of the border between those two topologically associated domains, is leading to the boundary deletion leads to the reconstruction of the three-dimensional changes in the chromatin. Okay, so you see here that single deletion over here actually leads to the fusion of two independent dots, dot 3 and dot 2. Now we have only single dot 2 composed of initial dot 2 and 3. So we see the changes in 3D structure, and those changes in 3D structure actually leads to the unexpected interactions between enhancers and promoters, therefore activation of genes which were supposed not to be activated. So indeed, we can confirm, and this is done by Lupanes with colleagues, that such deletions can lead actually to a lot of functional changes. And this was done by Lupanes with the deletion of the boundaries of dots, actually over here. But we also, in 2015, we performed analysis, and we observed that even a single SNP can affect the chromatin loop, presence by prohibiting binding of CTCF. Okay, chromatin looping, short recap before we will go to the computational methods. Two main sources of chromatin looping data. First one is based on a High-C, published in 2014, and the chromatin loops are those bright spots in the heat maps. Whereas the data which we are using mostly is based on the Chiapet, so this is the interactions I was telling you, CTCF or RNA-POL2. And what we observe is that CTCF seems to be, those interactions seems to be responsible for the chromatin architecture, whereas RNA-POL2 interactions are more dynamic. They are forming for a shorter period of time and then reassembling, drawing genes and enhancers towards so-called interaction centers. So it's like condensation of the phases in the system. Actually, here is one phase, namely the interaction center recruits multiple proteins and also multiple fragments of DNA, chromatin loops, by RNA-2 chromatin looping, on top of the predefined and designed based mostly by the because of the sequence CTCF chromatin looping. Okay, the modeling. There are different approaches which can be applied. Either you can use polymer physics model, in which you de novo introduce certain constraints and then construct the 3D models, and then you can calculate the observables. Or you can perform data-driven modeling, when you use directly the experimental data and then you construct the 3D models which are in agreement. And also the final methodology, which was introduced by Di Piero and colleagues, and 1 year later we also introduced this methodology based on epigenomic marks. You can actually predict the 3D structure without using. So this is somehow in between the data-driven, where the data is actually 3D context, and the polymer physics, where the input is not exponential data. So our methodology is so-called 3D Genome. I will maybe not go too deeply into details. The input is actually the triplet experiment, so singletons and pet clusters. Then we perform certain steps of denoising and preparation of the interaction data. Then we generate the 3D models, and then based on the 3D models, we construct the ensembles actually of them. So we tested our approach for multiple cell types. So for example, for GM12878, we will have around 80,000 of the CTCF loops, and if you actually requires Cohesin peak to be present, then it's around 40,000 of chromatin loops, whereas RNA-2 loops would be of the order of 70,000 such loops. And then you can, based on this experimental data, construct the 3D model, and the construction is founded by the polymer physics, and it's top-down approach. So you start first with the localization of chromosomes in the nuclear space, then each chromosome is being modeled as a chain of TATs or CCDs, those interaction centers I mentioned earlier, and then each CCD or TAT or interaction center is being modeled precisely as a collection of chromatin loops using some polymer physics model. So from the input data, so CTCF interactions and RNA-2, taking into account the orientation of CTCF motif, then we can construct the 3D structures for the interaction centers or TATs. This is one of the visualizations. Actually, what we are constructing, and each spatial scale, we're constructing the ensembles of 3D structures, and those ensembles of 3D structures then can be analyzed in terms of building, for example, the heat map, which reflects the probability of interaction. You can test your modeling with the noise by performing noise analysis, by adding noise basically to your data. And this methodology is actually presently available as a 3D Genome 2.0 web server, which do additional things. So from sequence, it is able to predict the 3D structure. And based on 3D structure, you, as a biologist, you can identify and understand better the biological function. Okay, so the web server is accessible under this link, and the 3D architecture of the whole service provides actually needed computational resources to actually perform such multiple jobs. Okay, so I will stop in a moment. The last slide I would like to share with you is actually related with the modeling engine, which allows in a very precise way. So here there is a tau region of the human genome and a specific set of interactions. the slide which shows how we do the 3D modeling across the population. So based on a reference 3D structure, and the chromatin looping, which is identified by the experiments chiapet, in this case CTCF and RNA-2 chiapets, each structural variant, so the change of the DNA sequence, is being mapped onto the reference 3D structure. And if, for example, by the deletion, you remove the C segment, so the anchor which harbors two chromatin loops, then basically those chromatin loops vanish and you have changes on the schema, in this case, schematic 3D model from the model which has all four chromatin loops to the model which has only two. Okay? And it's then modeled by polymer physics into the 3D structures, actually ensemble of those 3D structures. And similar for duplications, inversions, insertions. Okay, so that's our idea. That's how it works. Maybe I'll stop here, and I will just put a summary. So with our in silico technology, we are able to model 3D structures across the whole human population. So as an input, we use only DNA sequence. But in addition to the DNA sequence, we have to use reference 3D structures. In this case, it was GM1287, so it was lymphoblastid cell lines, B cells, and therefore the 3D models across the human population will be valid only for B cells. Okay, that would be not 3D structures for neurons or some other types of cells. So I would like to thank founding agencies, Foundation for Polish Science, Food by Fellowship, also European Union Empathy Project, and many other sources. And of course, I would like to thank also my team, my laboratory, a lot of people involved in everyday work and also with the results. I would like to mention Michael Sadowski, who was the first author and actually very active across the whole project, and Przemek Szałaj, Zofia Parteka, Michał Łaźniewski, Agnieszka Kraft, Michał Wasnodolski, who was co-author of the Predominant 2.0, the main author actually, Michael Kadlew, and our collaborators, Jimmy Wang, Charles Lee, collaborators from Poland, and also Celia Moy from Zurich, who helped us a lot with calibrating our MGS-based 3D computing methodology to using the high-resolution imaging data. Okay, so thanks a lot. This is our workplace Center of New Technologies at the Warsaw University, and this is the Warsaw University of Technology, the Department of Mathematics and Information Science. So thanks a lot. And I'm welcome to answer any questions you have. Please.
Thank you, Professor Flinski, for your talk. Are there any questions in the audience? You can use the raise hand function in the participants section if you have a question. Don't see any yet. I guess I will ask a question first, since no one seems to have one yet. You said this was specific for B cells you're modeling. What extent do you think you can modify this for other cell types? I guess you have to get the high C data, and we know that the mapping will differ between cell types, and also with different chromatin modifications. How do you think that will affect your mapping algorithms?
We actually perform a specific analysis, and we are working actually in the last couple of months on doing the chiapet experiment across different individuals. So in that way, on the same cell type, so in this case, B cells. So we can actually compare the experimental data, three-dimensional experimental data across different individuals for the same cell line. And it seems that the core of interactions are being present across individuals for the same cell type. And of course, then the next step, which we are currently working, is to go across different cell types with the first chiapet as our source for the modeling. So the source, the list of interactions, both CTCF and RNA-2. And we are currently working on 4 cell types comparing them, and definitely we see a lot of differences between cell types. Okay, so that's why we believe that the ideal experiment would be to do the ChIAPET or high chip or plug-seq. I mean, any type of the technology which gives you chromatin looping for CTCF mainly and RNA-POL2 across single individual multiple cell types, and then with this technology I presented today, you could then scale up the models for the different cell types across the population. In our recent paper, we actually compared high chips for CTCF and RNA-POL2, and it's across individuals. So they also seems to be very highly correlated. Okay, so that's...
Yeah, no, great. Thank you. The next question, I think, is from Jose, if I'm not mistaken. I only see...
That's right. That's right.
Okay, thank you.
Okay. Here's Jose Onushik. This was a very nice talk. I really enjoyed it. I think we all have been interested in modeling chromosomes and the full nucleus. I'm interested in the idea that going from a single chromosome to the full nucleus. You said that you have this hierarchical approach, appears very interesting and very promising. But I always wonder, are we allowed to go hierarchical? Are these energies in the same level that bases hierarchy, although it's conventional in terms of computation, may not be true in reality, that the interactions between... So do you think really the interactions between the chromosomes are weaker than inter and intra that we're allowed to go hierarchical, or that's just a convenience?
That's a very good question. So to be honest, the ChIAPET, but also other techniques are more tuned to see interactions within the chromosome. So those interactions will be like visible more strongly. In our approach was somehow trying to provide a certain solution for biologists in terms of the very rapid construction of 3D models. So it is done in the way that actually the modeling of the whole nucleus can be done within seconds or minutes in the browser. So all the code is like embedded in the browser. So you can model almost instantly the 3D structure. The only way to do it is to go from top to down, not other way around, because otherwise the complexity of computations will grow rapidly. Regarding the differences and similarities of the modeling parameters and also the details of the force field, I would say that at least from our, but also other work, it seems to be clear that those interactions which are below 50 kb with the resolution, so like 1 kb, like in our case, ChIAPET, seems to be governed by slightly different physics of, certain proteins bindings, and whereas the scale which goes like over 50 kb, 100 kb megabases, that would be maybe driven mostly by the epigenomic marks and certain slightly different force fields. So in our case, in our model, we actually optimize each scale separately. But typically the difference is, between the chromatin loops and like coloring of the chromatin fiber in the scale of domains and chromosomes. So that I would say that, the border, for example, one additional argument for that. So we perform a machine learning, deep learning model. We train it to recognize the interactions. So the model which works very good in the scale of below 50 kb is actually not so good in the scale over 50 kb, and vice versa. I mean, if you take the model which is trained over the 50 kb, which would be one of your actually papers I mentioned in the talk, then the question still is open. I mean, how to predict very precisely the chromatin looping. So there is like, that's why I think it as a kind of...
No, this is very nice. I'm not criticizing. I'm just wondering, in a sense, we all try to do this modeling when you're allowed or not allowed to do this sort of partition. That's very convenient computation. I'm not sure that physically it's correct, but that's a very nice answer. Thank you.
Yeah, I cannot agree more.
Great. So the last question is from Peter.
Yeah, that's very nice. And very ambitious to get everything down from the big scale to the small scale. I was wondering, when I looked at the few structures you showed, I'm afraid I never got a chance to study your structures in general, but in the pictures you showed, they looked very intricate. And I wondered whether there are knots in the structures that you find.
So the structures could be accessed in our web server, but I should mention that this web server is somehow in response to certain needs of biologists who would like to see some, some spatial details, but not putting too much of certain details which make the picture more fuzzy. So for example, the biologists tend to use our average structures, and every time we try to do some specific analysis, then when we provide ensembles or we start to calculate some statistical physics observables, it's for them it's of course very interesting, but for the purposes of the publications or even to discuss certain things, they prefer to have certain simplification. So please keep in mind that the certain things are actually with the purpose of visualization in mind, and it's maybe not political to admit, but most of our users of our web server are actually biologists who prefer to have very clear single average structure with the distances when they can measure in Chimera or some other softwares and play with that.
You're probably in touch with Joanna Solkowska. I don't know if she loves knots. So if you work with her, I think you'll find some very interesting things.
Definitely, anyway, that's something which we are actually currently working.
Okay.
And I would say that it's very tricky. So we actually developed with the help of mathematicians from the Warsaw University, our collaborators, algorithm which take the set of contacts and predict if the region is not knots or links, what's the topology of it. And I would say that the results at the scale of chromatin loops are rather discouraging in terms of density or number of nodes which are occupying genome. And of course, we know from biology that topoisomerases are actually releasing those topological stress by cutting and then allowing to equilibrate the complexity. Still, we were counting that for the idea that actually each topologically associating domain is somehow centered on the specific set of links, which somehow keep the chromatin somehow under the certain topological constraints. But it seems that it's more complex than we thought. Of course, we are working on the population averages. So, this is actually a tremendous question, and working with the single cell data, 3D genomics data, it's still not at the scales we would like to discuss. Okay.
Thank you.
Yes, thank you, Professor Polinsky, for your talk, and everyone should say thank you to him. And our next talk will be Megan King from Yale University, and she'll be just showing us a nucleosome constrained model for topologically associating domains.
So it's a pleasure to be remotely here today to share a recent story that is really come from a really fun collaboration between my group, and I'm in the cell biology department at Yale, and the group of Simon Mokri, who is a physicist at Yale. And I'm just showing up front here the pictures of the amazing team who have contributed to this project and really worked hard to bring together our biological data, which is predominantly my contribution to this work, with simulations that have been carried out in Simon's lab. And also, just before I begin, I just want to point out that the work that I'll be describing today was recently deposited on the bioRxiv, and so you can go back and look more at the data there if you're interested. And so I just want to start by introducing everybody to the concept of TADs, or topologically associating domains. And TADs over the past 10 years have become really foundational in our understanding of the kind of mesoscale of chromatin organization. And TADs are seen across all organisms. So what are TADs? TADs have really been revealed by techniques like Hi-C, and I'm just showing some contact probability maps or interaction maps that come from Hi-C data, where you can appreciate as you look down the chromosome, there are regions of relatively high contact probability, and then there are boundaries that segregate these regions from upstream or downstream TADs, which again have higher contact probability within these topologically associating domains. And in particular today, I'm going to show you biological data coming from one of our favorite genetic models, and that's Schizosaccharomyces pombe, which is a fission yeast. But in our simulations, I'll also be talking about work where we've leveraged Hi-C data from mouse. And that is helpful to us because these topological domains are much larger than those that are seen in fission yeast. Another key player in my talk will be the SMC complexes. So SMC complexes, like TADs, are broadly conserved from prokaryotes to eukaryotes, and they all have this type of architecture that I'm showing you here of these rings. And in particular, I'll be focusing on Cohesin and Condensing, which are two flavors of SMC complexes. And there's one form of Cohesin and one form of Condensing present in Schizosaccharomyces pombe. Again, that'll be the data that I will be showing you. So one thing that's very clear is that not only are SMC complexes like Cohesin and Condensing contributing to the topological domains that are observed in vivo, but also there are very interesting relationships between their occupancy in the genome and these boundary elements that are seen between topological domains, and I'll come to that in a bit. So my lab has long been interested in what determines the dynamic properties of chromatin, and we approach measuring chromatin dynamics using the approach that I've outlined here. So we take lac operator sequences, which are derived from bacteria, and we engineer them into fission yeast. We can then visualize these chromatin tags by expression of GFP fusions of the lac repressor lac-I. And this allows us then in the microscope to be able to monitor the position of a region of the chromatin over time. And so here I'm showing you an example of a field of fission yeast. You can see that each of these is an individual cell, and there's one locus that's lighting up here per cell, and that position of that depends on where we have inserted the lac operator into the genome. We can then take movies of the motion of these chromatin loci over time, as I'm showing you here, and we typically, for the movies I'll be showing you today, we've extracted data from the 50 millisecond timescales. So how do we go about taking this kind of dynamics of chromatin and extracting quantitative information? And so we do this by following the position of these lac operator arrays over time, as is depicted here. And one nice thing about lac operators is that they are diffraction limited, and so they're really well described by the point spread function of the microscope. And this allows us to reconstruct the position of the lac operator with high precision. And then we can track these particles over time, and I'm just showing you an example of what a particle track would look like. And then from this particle tracking data, what we do is that we convert this into an expression of the diffusivity of the chromatin, specifically the mean squared displacement versus time. Now, this is something that has been done in many organisms over many years, and typically what we observe for chromatin dynamics across organisms is an MSD plot, similar to what I'm showing you here. This is what we consider kind of a generic locus in fission yeast. In this case, the operator is near the gene MMF1. So if we were to look at kind of normal diffusion, what you'd expect is a straight line in an MSD versus delta T plot. But instead, what is commonly observed for chromatin is this constrained diffusion. And so you can see that instead of being linear, that over time, the motion of the locus falls off. And this can be described by a scaling exponent. And though I don't have a lot of time to talk about this today, we have determined the scaling exponent that's appropriate for fission yeast by fitting data, such as the data that I'm showing you here. And that scaling factor is between 0.44 and 0.45. And more information on that aspect of this work is available in the preprint, but I don't have time to talk about it today. So we observe a characteristic diffusivity on this time scale, and I just want to point out this is the seconds time scale for an individual locus like MMF1. But really interestingly, if we look now at many different loci, so here I'm showing you now 5 additional loci that are at different positions throughout the fission yeast genome, we see that actually the diffusivity is very similar, again, on the seconds time scale. And this might be different if we were to look at the minutes time scale, where other kinds of constraints to chromatin mobility might come into play. But on this time scale, we actually see a very characteristic behavior. And again, from this kind of behavior across different regions of the genome, we're able to describe this scaling exponent. So one of the other mysteries that has long been appreciated for decades at this point is that the motion that we observe for a chromatin locus in a live cell is dependent on energy, at least to some extent. And I'm just showing you an example of what this looks like in fission yeast here. So again, here is that MMF1 locus that I've already described. If we now deplete energy by the addition of sodium azide, you can see that this has a really profound effect on the observed chromatin mobility. And the diffusivity drops by about 50%. In addition, in this plot, we just have cells that are fixed, just to convince ourselves that the motion that we observe here is true motion of the chromatin mobility. So the source of the energy that drives chromatin mobility through the use of ATP has long been a mystery, and it's one of the things that we were hoping to address in this study. So one of the things that was an obvious candidate, particularly in fission yeast, is a role for the cytoskeleton. So in fission yeast, the chromosomes are organized in the so-called Rabel conformation, where the centromeres are tethered to the spindle pole body, which is the centrosome equivalent in yeasts. And then the chromosomes are kind of splayed out because the telomeres are also associated with the nuclear periphery. And then there's often the constraint of the ribosomal DNA, which is in the ends of chromosome 3 in fission yeast in the nucleolus, which is depicted here in red. So in fission yeast, this spindle pole body interface is actively driven by microtubule dynamics. And so microtubule forces are being exerted on the centromeres of the chromosomes. And so we were interested in whether this might be one of the contributions of energy to the chromatin mobility that we observe. So I'm showing you two different loci. So MMF1, again, our kind of favorite that I've already introduced, is shown here in red. Just notice this is a different scale, and that will become apparent in a moment why. And so you can see that when we disrupt these microtubule forces, which we do by the addition of this drug MBC, then you see that there is only a very small effect on the motion that we observe, particularly on these short second time scales. And so that's reflected by a very small decrease in the diffusivity on this time scale. What I'm showing you here in cyan is a lac operator that is instead inserted directly into the centromere region. So we're really looking at the centromere dynamics. And here on this short time scale, what you observe is that actually this is one region of the genome where the chromatin is more constrained than those 6 loci that I presented to you before. Again, here, if we depolymerize microtubules, this has very little effect. And that's because here we're really looking at a time scale that is shorter than the time scale of the driving of the microtubule dynamics. If we now look at longer time scales, so here you'll see that I'm showing you for the tens of seconds, what you'll instead see is a really different behavior for MMF1 and the SEN2 lac operator. So on these long time scales, you can see that there is an influence of microtubule dynamics on the diffusivity that we measure at MMF1, but that actually the centromeres which are actually driven have a very different profile. And this reflects the active driving from microtubules that we would expect to see. So this greater or super diffusivity is because of the energy dependent driving of the centromeres by microtubules. So taken together, this argues that microtubules are not having much of an effect on the time scale that I'm focusing on today, which is the seconds time scale. And so this does not appear to be really a major contributor, even in fission yeast, to the behavior that we're seeing for chromatin. So what might be contributing, right? So if it's not microtubule dynamics, what other ATP-dependent activities might be responsible? And so one of the things that we are really interested to explore is the potential role for loop extrusion. So let me introduce this loop extrusion factor model. Much of this has been developed in the lab of Lee and Mirny. And what I'm showing you here are some simulations of this model that they have carried out. So the concept of the left model is that loop extrusion factors, and loop extrusion factors we can equate with the SMC complexes that I introduced at the beginning of the talk. That loop extrusion factors are randomly loaded onto the chromatin, and by holding two regions of the DNA together and then extruding the DNA through the complex, this leads to the formation of these loops. And so that's the so-called loop extrusion activity. And the idea is that there would be continued loop extrusion until these loop extruding factors reach boundary elements, which are shown here in red in this diagram. And one of the most commonly thought boundary elements to be important for the formation of topological domains is the factor CTCF. And so that's one kind of biological molecule we can imagine, but there are also many other factors that likely contribute to boundary elements in the genome. So once these loop extrusion factors arrive at these boundary elements, they can no longer continue to extrude, and this would lead to a more stable loop conformation that still is dynamic, because these loops can still fall apart and be formed once again. So if we then look at what we predict for the position of topologically associating domains, what you can see is that the boundaries between individual domains are predicted by where these boundary elements are found. So the loading of the left is random, but then the left is stopped from extruding further by these boundary elements, which are embedded in the sequence of the DNA. So this was an attractive possibility for another type of activity that could be driving the chromatin mobility that we see that is energy dependent, because these lefts, cohesin and condensin, are ATPases. So this is the model that I've already described. And importantly, there's been a whole host of really beautiful studies demonstrating the ability of SMC complexes, cohesin and condensin, to use ATP to drive loop extrusion in an ATP-dependent manner. And so this led us to ask, is it cohesin or condensin activities that are actually driving the chromatin mobility that's dependent on ATP in fission yeast? And again, fission yeast is a really nice model because we have alleles that are available where we can critically inactivate these SMC complexes. And that's what I'm going to show you here. We took advantage of several alleles. Here I'm showing you a temperature-sensitive allele of Ms4, which is a loading factor for cohesin, and one of the SMC components, a cut14 of condensin. And so what was very clear when we looked at these critical loss of function by switching temperatures of these alleles and looked at their effect on dynamics is that they did not, that the data did not conform to what we had predicted. So here again, our favorite locus, MMF1, you can see that if we inactivate cut14 or Ms4, what we actually see is a boost in the chromatin mobility. So this is inconsistent with the idea that the activities of these factors are major drivers of chromatin motion. Instead, we observe more motion when we inactivate these components. This is not specific to MMF1. I'm just showing you another locus, PFL5, where we observe the same type of increase in chromatin mobility when we inactivate cohesin loading or condensin. So this data really suggests that the predominant effect of SMC complexes on the chromatin mobility is constraining. And that constraint we can think about because of the looping interactions would be constraining to the chromatin mobility, and that the loss of these looping interactions will lead the chromatin to become more mobile. And so while this is really interesting, it also didn't help us to understand, once again, what are the factors that are actually driving this ATP-dependent chromatin mobility. And so one thing we came back to considering is the potential influence of nucleosomes, right? Keeping in mind that DNA is not naked in the cell, but in eukaryotes, it's going to be packaged by histones into nucleosomes. And some very nice work from Eric Green's lab had already demonstrated that if you look at cohesin in vitro loaded onto DNA, that nucleosomes can have a profound effect on the dynamics of those cohesin molecules. So they can kind of diffuse along the DNA in the absence of nucleosomes. But when single nucleosomes are inserted, which are shown with the asterisks in these plots, there's a depletion of the residence of cohesin at these positions. And indeed, on a fully chromatized substrate, the cohesin is highly constrained. How this would work kind of depends on what we think about the structure of the ring of the SMC complex relative to the scale of the nucleosome. And I just wanted to point out that some recent structural biology really suggests that the ring could be more collapsed maybe than was first thought, which would be consistent with the nucleosome being able to prevent the diffusion of cohesin along the DNA in this type of experiment. And indeed, work from Frank Ullman's lab over the past decades has demonstrated biologically that this is likely to be important when we consider where cohesin molecules are found in vivo. This is looking at the RAD21 component of cohesin by chip data. And what Frank's lab has long observed is that the molecules of cohesin are often kind of piled up at regions of convergent gene transcription. And that has suggested that there is a constraint of the position of those cohesin molecules that is retained at these sites of convergent transcription because there is no further nucleosome remodeling that might allow for these molecules to move. And this has led to models such as the one articulated here by Frank Ullman with the idea that that SMC complexes are also preferentially loaded into nucleosome depleted regions, which are often found at gene promoters, and that these SMCs, particularly cohesin, can only move along the gene coincident with gene transcription, and then the molecules are dumped off at the site of termination, again leading to cohesin accumulating at convergent genes. And the idea is that this is dependent on the long lifetime of cohesin, whereas condensing is more mobile, and so it may be less prone to this. So this led us to ask, do nucleosomes present a barrier to left translocation? And do nucleosome remodeling contribute to the chromatin dynamics that we've seen? This leads us to additional factors. Indeed, this is supported by data from Susan Gasser's lab, where they found that driving trilocal transcription can boost the chromatin mobility. This is now in budding yeast. And this actually depends specifically on the Inouye chromatin remodeler, but not on SWR or RISC. And using an approach where they could target the Inouye complex to a locus, they were able to show that just targeting Inouye was sufficient to boost chromatin mobility independent of transcription. So we followed up on this by looking at nucleosome remodeling in fission yeast, and our data largely recapitulate what the Gasser lab described in budding yeast. So here again is our locus MMF1. We find if we deplete the protein ARP8, which is a non-essential component of the Inouye chromatin remodeler, this does lead to a decrease in chromatin mobility. For reference, here is ATP depletion in gray. If we deplete ARP9, which is a component of SWR and RISC, we don't see any effect on the diffusivity. So again, Inouye is somehow special, and I would just point out this is actually a hypermorph of Inouye because Inouye is essential, and we're very interested to determine using oxide-mediated degrons if all of this mobility is in fact due to Inouye activity. We wanted to combine this with loss of SMC complexes to really interrogate whether these are acting together, and so far these have been our observations. So again, if we look at a loss of condensing function here, you see that the mobility is boosted. We can see that the mobility is now further constrained if we also deplete ARP8, but the mobility does not go down to below wild type levels. And so this suggests that there is a complex relationship between SMC complexes and ARP8. And this is, and if we look at the permissive temperature, really, we can phenocopy what we see for an ARP8 knockout, even in the presence of CUT14, which you would expect because the protein is functional at that temperature. Okay, so I've already introduced this concept that transcriptional units might be important for the movement of SMC complexes through nucleosome remodeling. So this led to a prediction, and it led us to ask if the loading and steady state distribution of SMC complexes isn't beholden to this underlying gene architecture. So we asked, can we actually predict the folding of the chromosome, these topologically associating domains, just based on the position of genes? And so we built these simulations, and so I'll just take you through the concept of the simulation. Here we have bias loading of SMC complexes at gene promoters, which are the most common type of nucleosome depleted region. Those SMCs are constrained to the promoter Unless there is remodeling of the nucleosomes that are adjacent that would otherwise constrain the SMC. This would be coincident with the transcription, and so it's directional from that promoter through the gene itself, which would lead to the eviction or remodeling of nucleosomes that would allow the SMC to slide. And then rebinding or stabilization of nucleosomes in the wake of the replication, of the transcription machinery would then lead to the rebinding of nucleosomes, leading to the trapping, essentially, the SMC complex in a new position. And that this would go until reaching the end of the gene and termination of transcription. And then these SMCs, again, are dynamic and might dissociate. So we modeled this, again, now we decided to turn to the mouse genome because topological domains are larger, and so there's more data off of the diagonal. Here, I'm showing you experimental data in the top quadrant and in our nucleosome constrained model on the bottom left. And I'm showing you just the distribution of genes, which is the information that's been used in our simulation, but I'm also showing you the distribution of CTCF sites, which is important for other simulations that have been carried out. So using just the information about the position and the strand in which the genes reside, what I hope you can appreciate is that we can predict a number of elements that are seen in the experimental data. Here, for example, is a very strong boundary that is predicted based on our simulation and is observed in the experimental data. And you can see that in general that we predict a number of topological domains along this region, which are reminiscent of what's seen from the experiment. We compared this to, again, models that have been developed based on CTCF in the Mirny lab. So these models are instead based on the distribution of CTCF as described by ChIP-seq across this same region of the genome. And you can see this also does a very good job of predicting where topological domains will be found. So I just want to point out these two models are using entirely orthogonal data, but actually do a reasonably equally good job of predicting the local topological domains. And in fact, because of that, we can actually combine these models together. And what you can see is compared to our nucleosome constrained model, that the addition of CTCF really enforces stronger boundaries. I also just want to point out there are regions of the genome where the CTCF-dependent model fails to predict a boundary, as shown here with the arrow. And this makes sense because there is no CTCF in this region. But we actually predict that there is a relatively permissive boundary in this region when we look at our model, which is based on where the genes are located. Okay, so the take-home message is that genome architecture alone, just the position of genes within the genome, can predict TADs and boundaries. These strong boundaries can be predicted equally well in many cases by this model or basing a model on where CTCF is accumulated. I want to point out that in the mouse genome, this is not as simple as the data for budding yeast, where boundaries are predicted to be at convergent genes. It's actually much more complex than that. In fact, intuitively, just by looking at the gene position and density, we can't really predict by eye where strong boundaries will be predicted by the simulation. And so we're still trying to understand this. Thus far, our best guess is that there are regions of high gene density next to relatively gene deserts seem to be something that often leads to a strong boundary being predicted. And this could be because there's a high degree of loading of SMC complexes at all of the promoters of those genes, but that a gene desert would prevent the translocation of SMCs through the adjacent region. And lastly, one thing that we think is very interesting is that because we do predict many of the same boundaries with either the nucleosome constrained model or with the CTCF model, that there's an interesting relationship evolutionarily between the reinforcement of these two potentially independent activities. And for us, this is also important because fission yeasts don't have CTCF, and so we need to understand how TADs can arise in the absence of at least CTCF-dependent boundaries. So I've proposed this nucleosome constrained model. We argued for an important role for gene organization and predicting TADs, and we can do this now for genomes where we actually don't have any high-C data. And just a last food for thought is that our model suggests that non-coding RNAs could actually be important for remodeling topological domains, independent of any function of the transcript itself, which is something that we're interested in pursuing. So with that, I just want to thank again this fantastic group that I've already introduced, my collaborator Simon Mokri, and funding from the NSF through the EFRI program, which has funded this work. And you can find out more about this online at the bioRxiv. Thank you very much.
Thank you, Professor King, for your talk. Again, if anyone has a question, please use the raise hand function in the participants section to let me know that you have a question. Professor King, where are you?
I'm here.
Okay, cool. Great. So the Inouye Mirni has the first question.
Hi. So I wonder what's the general take of your model as far as the relative strength, relative importance of CTCF versus transcription? Because clearly you see some boundaries at transcription, but at the same time, CTCF certainly plays a huge role because CTCF removal leads to largely disappearing of most of the features, not all of them. So how would you weigh the two relative to each other?
Yeah, I think that's a great question. So for the simulations, I would say, right, it depends on which region we're talking about. So one model quantitatively might do better or worse in a given region of the genome. And for many regions, as one of the ones I showed there, right, they actually really often predict very similar things, even right using entirely different information. So just to make the point that that it's not as if they're are not both able to predict the same region, which just suggests that there is an important role, because otherwise we would not be able to do as good of a job predicting, obviously, with where CTCF is located, right? So just to be clear that obviously this is playing an important role, certainly in the mouse genome. With respect to experiments that have been done by depletion of CTCF versus Cohesin, I think you do see kind of what I suggested there, which is that the strength of the boundaries likely are reinforced by CTCF because you'll get running on, and I think that's very clear from that data, where you get molecules, persisting through regions beyond the boundary. I think one thing that's challenging is that we annotate the genome with respect to gene units that are dependent on RNA sequencing data, which really only shows the mature transcript. But we know, for example, polymerase can keep going and often does keep going, and that's not really reflected in the RNA-seq data. So one thing I think will be important is one of the things will be important for interrogating this model is really thinking more about the active transcription and doing better work to look at nascent transcripts and where those end as opposed to where our annotated genes end, to really think about how these two things might be intersecting quantitatively.
Okay, so our next question is from Jose again. I'll come back to him. Shubham, are you on?
Yeah.
Okay, go ahead.
I'm here. I'm sorry.
Okay. Okay, well, you can go. Whoever wants to go first. Maybe Jose, you go first. Yeah.
Well, these boundaries appear to be very, very robust. And this was a very nice talk, the way we present it. And looks like you come with these different prediction methods and they give similar results. Do you wanna, is that just an observation, or do you wanna tell me the real origin of it? What determines the boundaries? Is that just a convenient way of doing a predictive thing, or can we have some mechanistic or some physical justification why all of them give similar predictions?
Yeah, so I think one of the things that's very interesting is that one of the things that's very different is in the CTCF model, the loading is unbiased and the boundaries are set by CTCF position. In this model, the loading is not random. The loading is directed to gene promoters, and we're just using that as a proxy for nucleosome depleted regions. We could equally and maybe even more accurately, the model may be more accurate if we were actually using data of nucleosome depleted regions directly. And so that's a biased loading, but we don't set where the molecules will stop extruding, right? So very different kind of approaches. So I think that what it's telling us is that there may be roles for both a bias in loading and biases in where extrusion stops, and those could both be contributing, and also where they're not fighting each other. So they could have arisen, as I try to suggest, since many organisms like simple eukaryotes do not have CTCF. So there have to be other mechanisms that give rise to these boundaries. So with regards to what makes a strong boundary versus a weak boundary, as I said, the simulation with the parameters that I described does quite a good job at predicting many of these strong boundaries. But intuitively, and as a biologist, I'm not satisfied. We don't actually understand. Although I do think that what it means if there's bias loading is that there's a bias in the density of loading across the genome. And so this may be one important contributor. Depending on local gene density, that would be.
Great. Shubham?
Yeah, hi. So we do know that the kind of histone and DNA modifications that are present in the actively transcribed parts of the genome are very different from those present in, let's say, the gene depleted parts of the genome. So do you think that is also providing some sort of complementary information that is contributing towards the formation of TAPs?
Yeah, I think that's really interesting because while we think of the modifications that are going to make a region permissive to transcription, right, we also know that active genes are enriched in H3K36 methylation, which actually makes very stable nucleosomes in the wake of transcription. So transcription is interesting in that you can be making things more permissive on the front, but actually in the back, you're actually stabilizing nucleosomes. So there may be both of these flavors, which could be more or less stable than what you would find in a region that's not transcribed. So I think that's very interesting, but is more sophisticated thus far than on the kind of first principles of what we've been modeling.
Okay, thanks very much.
Thank you.
Great. So thank you, Professor King, again. In the interest of time, we'll move on to the next talk. The next talk will be from Sumat Brahmachari. I've been told to tell you guys that unfortunately there was some issue with internet connectivity during the talk, so the first minute or so has been cut. So there's no problem if something looks a little bit funny at the beginning of the feed when it starts. And the schedule also says that we're supposed to have a 15-minute break, but we're not actually going to have a 15-minute break because there was a typo there.
Hello, everybody. I am Shumita Brahmachari. I'm a postdoc at Rice University at Center for Theoretical Biological Physics. And today I will tell you about interchromosomal organization via lengthwise compaction of chromosomes. A little bit of introduction. So DNA is the chemical substance that contains our genetic information, but as a physical object, DNA is a very long biopolymer. To get you some reference with numbers, the human chromosome 1 is about 10 centimeters long, that is packed within the nucleus of diameter 10 microns. So you can see that there has to be at least 1,000-fold compaction in the linear dimension for chromosome 1 to fit inside the nucleus. However, volume-wise, the total volume occupied by all the genomic elements inside the nucleus is about 10% of the nucleus volume. So this tells us that the genome can be randomly packed. There's no problem in just randomly packing all the genome inside the nucleus. However, such a random packing can cause serious physical constraints during their mitotic individualization. Because these are long polymers, they can get highly entangled. And as a result, this individualization or resolution of individual chromosomes during mitosis can be a big problem. So it's not a surprise that the chromosome is not folded randomly inside our nuclei. Rather, there seems to be a hierarchical organization. To be more precise, the chromosomes are organized as a linear array of cis-interacting domains. And so which is what I mean by a lengthwise compacted polymer. So this is shown in schematic here. So these are these cis-interacting domains, and genomic elements within these domains actually preferentially like to interact within themselves rather than elements with other domains. And this leads to a significant degree of compaction in the genome, and as a result, and this also leads to a better mitotic resolution. And this is what the lengthwise compaction scheme is, that you have this domain morphology that compacts along the contour. Now, the details of these domains, the architecture of these domains is under investigation. However, chromatin loops are thought to be one of the major players of these domain interactions, and SMC complexes, or structural maintenance of chromosome complexes, which are condensins and cohesins, they are known to extrude loops and stabilize these loops. So these are active enzymes that can hydrolyze ATP and extrude these loops in a processive manner, which can lead to this formation and stabilization of these loops and maintain this lengthwise compaction of chromosomes during the cell cycle. There is another very interesting aspect of genome organization, which is the sticking of chromatin segments with themselves. So heterochromatin or euchromatin, depending on whether they house the inactive or active parts of the genome, are known to have some self-adhesion, and which drives their segregation into micro space-separated droplets inside the genome. In the figure here, you see very beautiful work where heterochromatin forms space-segregated globules. These bright spots are heterochromatin spots inside a Drosophila embryo. And there has been a recent study that proposes that the loop extrusion or lengthwise compaction actually counteracts this compartmental segregation that comes up from sticking of the polymers. Now, the idea goes as follows, that you have the blue and the red polymers, which are the two different types of polymers. In absence of loop lengthwise compaction, what happens is they like to stick to each other. They like to stay in separate compartments. However, this active mechanism of loop extrusion can bring the blue and the red polymers together, thus disrupting compartmental segregation. Now, this kind of a mechanism, one can imagine occurring in cis inside the chromosome for disrupting formation of compartments inside the chromosome. But how does this mechanism play out when we are talking about interchromosomal compartments is not clear. And to answer that question, we would like to, we developed a simulation model. So this model is based on previous work at our center, and it's very simple. We have 10 chromosomes inside a confined volume, and our chromosomes are a series of connected monomers or beads, where each bead roughly corresponds to, it can range from 10 to 100 kilobase of chromatin. So these beads, you can think of them as one domain or a couple of beads forming a domain. And in the chromosome, you have a centromere at the middle, and the centromeres represent heterochromatin that can self-adhere. They like to stick to each other. And each of these monomers or beads actually undergo stochastic Langevin dynamics with some thermal noise. And the force fields characterizing this Langevin dynamics actually are derived from a Hamiltonian, which looks like this. This Hamiltonian actually contains many terms. However, all these terms can be basically clubbed into three basic terms, essentially. The first term takes into account all the polymeric constraints and the logistics of actually maintaining chromosome as a polymer. By that, I mean all the nearest neighbor interactions, because the nearest neighbors have to stick to each other, and the next to nearest neighbor interactions, which has to maintain certain bond angle with them that controls the stiffness, and so on and so forth. And then the next term is the adhesion term, where centromeres like to stick to each other. When they come in close proximity, they will tend to stick to each other. And the third term, which is very important, is the lengthwise compaction term. And the way we implement lengthwise compaction, I will go into a little bit more detail later on. But in a few words, what this potential does is this potential acts along the contour, or the strength of the potential decreases along the contour. And whenever two beads, which are on the same chromosome, come near each other, and these are non-neighboring beads, they have a certain degree of stickiness, and the degree of interaction or the degree of stickiness will depend on how far they are in the genomic space. Essentially, what this term in the Hamiltonian does, what this force field does, is it increases cis interactions, increases the cis contacts in a chromosome. Okay, so once we have all these ingredients in our boiling pot of soup, what we have in our hands essentially is we have a sticky polymer, or we have 10 sticky polymers that are space filling, and we do allow chain crossing, but a bit with an energy penalty. So this mimics the in vivo scenario of topoisomerases. And most important ingredient is that these polymers can actually compact in a lengthwise manner. And what do I mean by that is going to be very important. What is the lengthwise compaction? How to interpret lengthwise compaction in our simulations? And what we are going to do basically is we are going to keep all these other terms fixed, and we're going to modulate lengthwise compaction levels of lengthwise compaction and see how the chromosomes behave and study the chromosome morphology and so on and interactions as a function of varied levels of lengthwise compaction. But before that, as I mentioned, we'd like to understand or interpret what does lengthwise compaction mean in our simulations. So this is essentially the energy as a function of interaction energy as a function of the genomic distance. And you see two curves. One corresponds to a weak lengthwise compaction. One corresponds to a strong lengthwise compaction, which is the cyan curve and the red, respectively. And the generic form of the potential is that it decreases as you go along the genomic contour, which basically translates into the fact that if you have SMC complexes that are extruding loops, smaller loops are going to be more stable than larger loops. As a result, the interaction strength is higher whenever two genomic elements that are close by come in contact than which are farther out. And the weak and the strong basically translates to a lower SMC activity versus a higher SMC activity, essentially meaning that for weak chromosome compaction, you can only form very small loops that are sparse, whereas for strong lengthwise compaction, which corresponds to higher SMC activity, you can form larger loops and compact the chromosome to a higher degree. So these are some representative snapshots of our simulations corresponding to weak and strong lengthwise compaction. What you see here are all the chromosomes in a transparent gray and one chromosome shown in blue. Whereas in these snapshots, all the different chromosomes, the 10 different chromosomes are shown in different colors. What you see is for weak lengthwise compaction, that is lower SMC activity, the chromosomes are spread out and they intermingle with each other to a higher degree. Whereas for higher SMC activity, you see the chromosomes form compartments or territories within the genomic space, and these territories are mutually exclusive. And this effect has been seen experimentally as well. So these are some nice experiments done where you see, this is the wild type case where you have different chromosomes occupying different territories, but as soon as you deplete one of the SMC complexes, that is condensing 2, CAPH2 is a subunit for condensing 2, the territory formation is lost, and this kind of effect we do see in our simulations as well. Now, I have not shown you any Hi-C map before this slide, so I am pretty certain most of you are familiar with what Hi-C map represents. But in a nutshell, what it means is it shows the interaction probabilities or interaction probabilities or frequencies between genomic elements in a matrix format. So these are both interchromosomal and intrachromosomal interactions. There have been shown that diagonal blocks are only intrachromosomal, where the off-diagonal are interchromosomal. And this is for for strong lengthwise compaction. This is for a weak lengthwise compaction. And what you see mainly is that if you have higher lengthwise compaction, the diagonal is thicker, representing more cis contacts between chromosomes and representing stronger territory formation. Whereas if you decrease lengthwise compaction, this is the interesting bit, is what you see that trans-centromere interactions, so these dots correspond to interactions between centromeres of two different chromosomes. They become more intense. And note that the intercentromeric adhesion between them, between these two cases, is exactly the same. So the only thing that is different is the lengthwise compaction term. So this is somewhat paradoxical, that a potential that a force field that is constrained within a chromosome, that is entirely an intrachromosomal feature, can modulate interactions that are interchromosomal. And I'll come back to this to answering this sort of paradoxical behavior. But before that, let me briefly mention that we do see this behavior in experiments as well. So these are experiments done by our collaborator Claire in Benjamin Rowland's lab at National Cancer Institute in Netherlands. And what she finds is that condensing 2 depletion actually leads to spatial clustering of centromeres. So what you see here is DNA in blue, and these dots are centromeres. Now upon depletion of condensing 2, this is a subunit for condensing 2, all the centromeres cluster. And the number of clusters basically roughly decreases by a factor of 2. Upon condensing 2 depletion. Now, to be perfectly honest, this study is largely motivated our study. So we can ask the question that whether we see this in our experiment, in our simulations or not, and the answer is of course yes. What we do here is basically we look at every snapshot and we assign clusters to centromeres, and we use various statistical methods to assign clusters, and all of them end up with the similar result basically. And what we see here is that upon increasing lengthwise compaction, the number of centromere clusters increases. So this corresponds to the wild type where you have many clusters. However, if you deplete condensing 2, the lengthwise compaction decreases and number of centromere clusters also decreases. This is a representative snapshot again showing the same. So why does it happen? What happens is lengthwise compaction is actually screening intercentromeric interactions. So these are a snapshot of one chromosome, and the red is the centromere and everything else is the arms of the chromosome. What you can see here is that the centromere is buried deep within the entire chromosome for high lengthwise compaction. Whereas for the other case, where the lengthwise compaction is low, the chromosomes are much more spread out and mixed with other chromosomes, and the centromeres are exposed to interact with other centromeres. This leads to a higher formation of formation of bigger clusters. And this, in principle, can act inside a chromosome as well for screening of genomic elements that are buried deep within one compartment or the other. This kind of screening can also work in cis as well. So with this, I would like to basically conclude. This is all I had to say. And so I would like to acknowledge my advisor, Jose, all my colleagues at Rice, and my collaborators, Michele, Erez, Olga, and Ryan, and as well as my collaborators in Netherlands, Claire and Benjamin, and the funding. And of course, thank you for listening. And I would leave up this summary slide, which basically summarizes what I had to say. It's basic 2 points: that lengthwise compaction drives territory formation, but enhancing cis chromosome interactions, which is a result of higher condensing 2 activity or higher SMC activity. And this lengthwise compaction can also screen transchromosomal interactions by burying chromatin segments within cis interacting domains. And that's it. Thank you.
So thank you, Dr. Ramachari, for the very nice talk. Yes, if anyone has questions, again, please raise your hand. Inouye has a question again. Great.
Yeah, so I was trying to wrap my head around. So basically what you've done, you replace loop extrusion with some effective equilibrium simulations.
Yes.
And you basically reproduce what we've done with loop extrusion and compartmentalization in this paper that you showed, Nucleosomes et al.
Mm-hmm.
What we also showed is that you cannot really fully capture the active process of extrusion by just lengthwise compaction, because if you just freeze the loops in place, you achieve this lengthwise compaction that you study, and it does not fully capture what loop extrusion does to compartments. Like loop extrusion has a strong effect erasing compartments than just fixed loops. So it sounds like you cannot really fully capture the active process by the equilibrium model. I wonder what's your thinking about this?
So yeah, that's right, that we don't simulate the active process. However, the idea is if you fix loops at various positions along the genome, we'll end up with different equilibrated structures. And using this potential, what we are doing is we are sampling all those different potential structures that you can have when you have different, when you fix loops at different sites, for example. However, this is one interpretation of this. This is a little more agnostic in terms of exactly what is causing this. But what I'm trying to show is this kind of a phenomenological parameter can in fact include this effect of if you have this polymer brush-like morphology arising from loop extrusion. However, not really fixing loops, but really looking at the statistics of when you have two cis points coming in contact, giving it a loop morphology. But it's a little more general than that in terms of capturing the morphology of chromosomes. So this is in that sense. But what you talked about is how loop extrusion is counteracting compartmentalization. So I was not exactly clear what you meant by that. What loop extrusion does when you have direct loop extrusion, it counteracts compartmentalization, but that effect is not really captured. So I was not entirely sure what was your thought behind that.
So you showed an image from our study as a motivation, the Nuclear et al. Paper. And so what this paper have shown is that loop extrusion, when active, counteracts compartmentalization. So compartmentalization gets weaker when extrusion is more active. And there are experimental evidence in support of this. In the Whipple knockout, when loop extrusion is particularly active, compartments get really weak. At the same time, we've shown that when cohesin is removed from DNA, compartmentalization gets much stronger. And so the model was the active extrusion fully reproduces this. And I would imagine that the question is, can your approximation of loop extrusion by just the effective potential achieve the same thing? Or not?
Oh, okay. Yeah. So
we've seen that just if you fix loops, that's not going to do what the active process does. You kind of do an active stirring of the soup with a spoon, sort of. Just not enough to put a spoon in the soup. You need to move it.
Right. I mean, so, okay, I understand your question. And the answer is yes, I think. So we don't have compartments in our chromosomes, right? In this simple model, you only have centromeres. So centromeres coming together is a form of compartmentalization, but this is trans-chromosomal compartmentalization. And when you see this lengthwise compaction, it's actually what it does is, as I was telling you, that it screens these compartment formation because you need to have these two come in spatial proximity, which is counteracted by this kind of contour-wise compaction scheme. So yes, in lengthwise compaction also counteracts compartmentalization, but in our scheme, this is basically intercentromeric compartments.
Thank you.
The last question is from Suhyo Shin. Sorry if I mispronounced your name.
That's fine. Hi, it's very interesting work. My question might be quite complementary to Leonie's question, because I might be interested in whether or not you got the POVs you would expect from the Heist experiment from your simulation. So have you checked out? Have you checked out the contact frequency? Plotted as a function of the the linear length of the genome distance.
Right. So we can do that, and I don't show it here, but I do have the curves that show. So when you decrease lengthwise compaction, the long tail is basically the power law decreases. So you don't have long range interaction. The long range interactions are decreased when you decrease this lengthwise compaction term. So I can extract that, and I have. So you're talking about contact probability versus genomic distance, the P of S, so-called PS of S, right? And it decreases with some power law, and that decrease is enhanced when you decrease lengthwise compaction in this model. Does that?
Okay, a very quick question. According to your model detail, I think that it's not self-avoiding polymer. The chain of crossing is a load. Does that affect your simulation result?
So
depending on the energy
penalty. So it is self-avoiding. However, two chains can cross with some. So there's an energy barrier to crossing the chains. You can make it infinite and make it completely self-avoiding, but we keep it around 4 kT, which allows some chain crossing. The chain crossing, it's not freely crossing each other. Yeah, but they are self-avoiding. So which I think mimics the Vivo scenario.
I mean, that is an important factor to to make the form of the P of S as well. So yeah, that's the reason why I asked this question. Anyway, we can talk later. Thanks.
Okay. Thank you.
Yes, thank you, Dr. Bhamashari, very much for your talk. We will now move on to our next speaker, Daniel Lee from Princeton. And he will be talking about chromatin mechanics dictate subdiffusion and coursing dynamics of embedded condensates.
Hi, everyone. My name is Daniel Lee, and I'm a fourth-year graduate student at Princeton with Cliff Brangwyn and Ned Wingreen. And today it's my pleasure to be talking to you about some work I've done recently on the effects of the viscoelastic nuclear environment, in particular the chromatin, on the subdiffusion and coursing dynamics of nuclear condensates. And so first I'd like to talk a little bit about the structure of the nucleus as a whole. And so at the broadest, simplest level, we have the major components of the nuclear lamina, which is sort of the structural outside of the nucleus, the chromatin, which is broken down into heterochromatin and euchromatin and takes up a very large volume fraction of the nucleus itself. And lastly, we have the nuclear bodies, including these nucleoli, these nuclear speckles, as well as a wide variety of other nuclear bodies, such as the PML bodies, the Ka bodies, and many of these are thought to perform crucial biological functions. Now, in general, these nuclear bodies are also thought to form by liquid-liquid phase separation. And while we understand how liquid phase separation works, and we've studied it for many decades, I don't think we properly understand the consequences of having a phase-separated system in a complicated viscoelastic non-equilibrium environment that is the living cell. And so my primary question is asking how this picture, essentially how the nuclear bodies in the cell differ from the simple physics that we see in synthetic systems such as this, which is available at your local Home Depot for not very much money. And so in the Brangwyn lab, we have spent a lot of effort trying to build tools in order to understand this question of how the physics of the living cell essentially deviates from the liquid phase separation that we see in that lava lamp, for instance. And so this is, here I'm showing one such tool that we've developed. This is called the Correlate System, which was developed initially by Dan Braha and colleagues. And it has in it we express these two components, the eyelid GFP ferritin, which multimerizes into a 24-mer ferritin core, we call it, and the IDR M-cherry SSPB. And so the eyelid and the SSPB are optogenetic partners. And so essentially when we turn on the light, they will bind tightly, and we will go from having this sort of core and IDR dissociated state to having a fully decorated core, as you see on the right. And now to this SSPB, we also have attached this IDR and this M-cherry. And so we will, these IDRs generate, or rather drive phase separation. And so when I turn on the blue light, as I'm going to show in a second in this movie, we'll go from having this sort of very diffuse pattern of fluorescence in the nucleus to having one that is very punctate, very sort of classically seen in phase separation. And so we can show that these puncta have liquid-like properties and so on and so forth. But one thing that we can readily observe is that these puncta don't really seem to change very much. So after those initial 5 or 10 seconds where we saw nucleation and the formation of these puncta, not that much has happened since we showed that. And so we can see how generic this phenomenon is. And in general, we find that if I watch these cells for hours and hours, here I'm doing it for about 2 hours with the blue light on the whole time, we actually see that the droplets don't seem to coarsen that much more. They essentially stay dispersed throughout the nucleus. And this is a little bit perplexing to me because in general, for a phase-separated system, we'll expect to see the formation of 2 continuous phases over a long period of time, like you would see here on the right. And so we can ask very readily, why don't we see one big droplet? Why don't we see the formation of 2 continuous phases? And so in order to look at that a little more carefully, we can quantify this, right? We look at the average radius as a function of the droplets as a function of time. And in general, we'll expect a power law such that average radius goes like T to the beta. And beta here, for a simple 2-component system, we would very naively expect a 1/3 exponent. However, if we do this over quite a few cells and then we perform the appropriate normalizations and averaging, we actually find that there is a beta of 0.12. And this, of course, is much less than 1/3. And so given this difference, we can ask, what is the mechanism, what is the physical basis of that deviation? And so to understand why this happens, we need to think a little bit about the mechanisms by which droplets grow. So in general, droplets grow by 2 mechanisms, right? So there is this thing called Brownian motion-driven coalescence, by which we have these 2 domains which will randomly walk and diffuse until they meet each other. And once they touch, they'll merge and form this larger droplet to minimize their surface tension. Likewise, Ostwald ripening is also driven by surface tension, but it doesn't require these discrete merger events. Essentially, a smaller droplet will be in equilibrium with a larger local concentration of monomer, then will a larger one. And so over time, there'll be this diffusive flux wherein we have movement of material from the smaller droplet to the larger droplet in a monotonic continuous fashion. And thus the larger droplet will grow continuously until it consumes the smaller one, and we end up in the same final state. And so in both of these mechanisms, we're driven towards the same two continuous phase sort of final state. And so in order to understand why they might be arrested, we need to look into which one is happening. So what actually happens in these nuclei. And in these nuclei, we can observe pairs of droplets that we know will merge. And so for instance, we have these two, which we can follow for minutes and minutes and minutes, and eventually they merge. But before that happens, they assume these two very stable sizes. They don't fluctuate a lot. Whereas after they actually do merge, they form this third droplet that's quite a bit larger, and that size is well explained by a volume conservation law. Of course, we can do this over many merger events and see that this is generally true, and that therefore we can conclude that the growth is going to always generally be explained by merger events in this system rather than Ostwald ripening. And so our mechanism that we've sort of concluded is driving the coalescence, or rather driving the coarsening in the system, is Brownian motion-driven coalescence. And so, the issue then is that both of these mechanisms, the Ostwald ripening, the Brownian motion-driven coalescence, they both give you a beta equals 1/3 scaling exponent. But we still haven't explained why we see this a beta equals 0.12 scaling exponent. But we go back and realize that, well, Brownian motion-driven coalescence, as it turns out, does actually assume Brownian motion, unsurprisingly. And this is quantitatively described by looking at the MSD, the mean square displacement of these particles, and noting that they will also be characterized by a power law with the exponent alpha. And so for perfect Brownian motion for normal diffusion, we will see that alpha is 1, and for what we term subdiffusion, we'll see that alpha is equal to 0.3, or rather less than 1 in general. And so here we have two simulations wherein in one we have an alpha equals 1, and one we have 0.3. And when I allow all the droplets to merge, what we'll see is that the droplets on the left will very quickly merge into 1 or 2 large domains, whereas the droplets on the right form many smaller ones over time and don't coarsen very much over time, in accordance with our observations from the microscope. And so once again, we can quantify this by looking at the power law of the growth as function of time, and we see that we can directly relate this coarsening exponent beta to the mean square displacement exponent alpha by a proportionality of 1/3. And this is, of course, actually borne out in simple analytical scaling arguments we can make, which are in the bioRxiv version of this paper, which is available now online, actually. And and so we might ask, though, well, we can explain the anomalously slow coarsening with anomalous diffusion, but why might we see this subdiffusion in a living system? Well, it turns out that subdiffusion is a major signature of a viscoelastic environment, and being that we're in the nucleus, our candidate for the thing that might be causing such a viscoelastic environment is, of course, the chromatin. And so we can look into how the chromatin interacts with these droplets, and to do so, we'll generally label the chromatin non-specifically, while also monitoring a droplet that we can locally write by shining blue light in a very specific location. And so when I turn on the blue light, we'll see, as highlighted in red, that where the droplet forms, there's a void in the chromatin. Essentially, the droplet must push out the chromatin, and when it vanishes, when we turn off the blue light, that void will sort of swallow up. And so in general, we'll note that these droplets can reversibly push out or exclude chromatin and embed themselves, and need to embed themselves in it when they form, when they nucleate. Summarized more quantitatively, we can see that if we look at the intensity of the droplet as radially, and we also look at the chromatin density in that area, we see that where the droplet is, the chromatin is essentially excluded. And so this is true, as it turns out, for many IDRs of choice, for many transcriptionally relevant IDRs, including your FUS, BRD4, TAF15, HNRPA, so on. And so, of course, then, given that we have a reason for subdiffusion, we can look into whether it's actually happening or not. And by performing single particle tracking on these condensates, we can actually see that there is significant subdiffusion around with an alpha of around 0.45 if we track these droplets over a long time. And so, of course, this does actually explain pretty accurately, based on our previous quantitative arguments and our previous observations of the coarsening, that why we have slow coarsening and slow growth of these droplets. It's also important to note that these behaviors of, in particular, the subdiffusion is actually very often known to be dependent on the size of the droplets. And so if we're looking at this, and so we can leverage actually the power of our optogenetic system and generate droplets of different sizes and interrogate whether these dynamics are the same and essentially over different over sizes. Essentially, we can look at the dependence of the mean square displacement on droplet size. And so we can, of course, do this once again. And we find that if we average over many droplets and look at many different essentially sizes, we can see that the mean square displacements all behave roughly similarly, and that they all have an alpha of around 0.5, which is consistent with a material that has a pore size far below our probe size. And so our minimum probe size is 130 nanometers, and so we can conclude that chromatin must have a pore size below that, which is consistent with back of the envelope calculations that we've done elsewhere based on electron microscopy and other data. And so this sort of highlights the power of this system in being able to describe the environment of the nucleus entirely based on these optogenetic probes. And so in summary, we've shown that these sort of embedded nuclear condensates form what I'll describe as an emulsion that's stabilized by chromatin. We demonstrate that the coarsening of this emulsion is based upon the properties of the nuclear environment, and in general, we've shown that these viscoelastic media can dictate the size and growth dynamics of emerging condensates. And finally, we've shown that these condensates may be used as microrheological probes of the nuclear environment. And so to close, I would like to thank my advisors, Ned and Cliff. I would like to thank both of my groups, both the Brangwyn and Wingreen labs, as well as my funding sources, and you for listening, and of course, the organizers for the opportunity to give this talk, and acknowledge also that this work, if this work is available now in full detail on bioRxiv, and I'm happy to take any questions online or on Twitter or elsewhere. Thank you very much.
Thank you, Daniel. Again, if there are any questions, please use the raise hand function. Daniel, are you here?
I'm here. Hi.
Okay, great. Yifeng He has a question.
Hi, Daniel. It's a really great talk, and I also check your papers online. It's a great paper. So I have several questions. Like the first question is like you have this. I guess this is more like a conceptual question. Like you have this artificial like granules that you can manually control it to coalescence into like smaller or big ones. Yes, I'm just wondering whether your artificial granules or like condensates would have any interference with the intrinsic condensates of the inside the cell, like the nucleolus of the cell itself.
Yeah. So in Cliff's lab, we've shown that depending on that, you have to be very careful about how these exogenous components interact with the intrinsic, as you say, membraneless organelles in the cell. So, for instance, if you look at our recent paper in Nature, Reback and Zhu et al., like Nature 2020, they look at optonucleoli. I think likewise, we've looked at how they interact with native stress granules. And so here in this particular paper, I've specifically chosen IDRs that are not miscible with that do not localize to like nucleoli or anything or any specific body. We've chosen like FUS IDR, which we have pretty well characterized and know that it doesn't overlap with anything that's already there, basically.
So basically, you're choosing something that could only strongly interact with itself. But
exactly. Yeah. So these are just so in the correlate system actually itself, we express the IDR and not the IDR truncation, which does not have which is not the full length protein, so it lacks any specific interaction to other specific things.
I see. Like if I want to ask a second question. Like I noticed your simulations. Like I just wonder, like you have like two cases. You have like a normal diffusion and subdiffusion case. I just wonder, like, it seems like in your normal diffusion regime, even in the normal diffusion, you still sort of end up with two condenses rather than one. I wonder whether that like, you know, like it's kind of it just kind of you know, little sort of surprising, because, you know, if you have like normal diffusion, you should end up, and you in your simulation you don't have like chromatin, I assume. Like you should end up with like a mono, like a single condensates.
Yeah, so that entirely depends on just the amount of time you run it for. Like in general, if you ran that for probably another 1,000 time steps or something, eventually those two droplets would find each other. It's just a question of probability of merging, right? And so essentially, if they move faster, they'll find each other more quickly. So if you run that for a little bit longer, you'll eventually hit one droplet.
And I also like wonder whether that also depends on your parameters of the simulation, like whether, you know, it definitely related your surface tension, right? Like if your surface tension is sort of too high, or like if it's too high, then it have stronger tendency to merge, I would say.
Well, yeah. So the way you would code that in is by looking at is basically by assigning a merger success probability. So in this particular case, if you ever merge, if the droplets ever find each other, then they immediately merge. But if you have a surface tension that's like sufficiently low, that will take some amount of time to force them to merge, or it'll actually have some probability of failing to merge.
Oh, so actually in your simulation, you don't have surface
Yeah,
tension.
this actually is just completely successful if they actually find each other.
I see. So
yeah.
Okay.
So if you just ran it, it's very simple. So if you just ran it a little bit longer, then they would actually merge. And
I
right, and so by forcing them to have progressive, like essentially the subdiffusion acts as the chromatin effectively in this very simple simulation. There are some more complicated ones that actually have like polymer networks and stuff, but that's sort of in the works with in the Wingren group.
I see. Thank you.
Yeah.
Great. Our next question is from Guangxi.
Hi, Daniel.
Hi.
It's very nice talk. Thank you. So I have a question. So it seems that the reason that the droplet does not merge all into one single phase is because this diffuse very slowly. So so I'm wondering. So when you what happened if you observe long enough? Does that, like, if you observe long enough, does it eventually merge to a single phase, or there some other mechanism actually can be in a way that the droplet cannot actually coalesce into a single phase? Yeah.
Well, to be entirely frank with you, if you keep the light on for like upwards of like 4 to 5 hours, the cell just dies. That's, to be frank with you, that's what happens. But if you were to imagine being able to do this indefinitely, like no cell division, like everything works forever, then like everything is fine and cell stays healthy forever, it would eventually, I imagine, merge into one large droplet, but that would literally take many hours. Like you're talking weeks, because the scaling behavior, right? Because the scaling behavior is so drastic, right? Like it's an exponent on the MSD, right? So if it's sufficiently small, then you definitely will. It'll take a very long time, and that's because you're sort of stuck in the chromatin, right? And so if you imagine being able to do it for a very long time, then it'll just go to one droplet. But it's really not physiologically possible, to be honest with you, in this particular system.
Okay, thank you.
Our next question is from Megan King.
So I'm just curious, why do you think that it seems thus far Oswald ripening is not seen?
So that's a super interesting question, actually. And there's a number of answers I can give to that. Number one is that the surface tension of these droplets seems to be rather small. And so in the calculation for the Oswald ripening, that goes in as a term on the numerator. So it's like directly proportional, right? So essentially, so observations from our lab on nucleolar protein in vitro give a surface tension on the order of 10 to the -6 to 10 to the -7 nanometers, rather newtons per meter. And that's also substantiated by in vivo studies done by Alexandra Zadovska's lab, which I believe we'll be hearing about later. And so that's all like low surface, that's all suggesting low surface tension. And so that would make the oswald ripening quite slow. Likewise, there is, well, in terms of something that's newer, there's a large body of emerging work in the soft matter community from Eric Dufresne's group, where they look at the mechanical inhibition, they call it, of oswald ripening by an elastic matrix. And so I personally would hypothesize that the same physics is happening here. And again, there's work in progress to sort of look into that more deeply. But yeah, super interesting question, and I think there's more there.
So just on that latter point, I was just curious whether you've looked at softening the chromatin, right? Because this is a perturbation you could do,
Absolutely.
which would be expected to affect the droplet size then.
Yeah, I totally would expect that if you can find a way to soften the chromatin wherein you increase wherein you can restore some amount of diffusion. For instance, actually, there's a body of work from Yuval Garini's lab suggesting that if you knock down lamin AC, then you can get diffusion of telomeres. So if you can do something like that, and then you might be able to restore normal diffusion and therefore get fast-growing droplets. So that's something else that can come, and that's also super interesting to me. Thank you.
Thanks.
Great. Guang, did you have another question, or is your hand still raised?
No, sorry.
No, it's totally fine. Okay, great. Thank you, Daniel, again.
Thank you very much.
And so for our last talk of the session, we will hear from Mateusz Szylinski from the University of Warsaw. And he'll tell us about consensus SV, consensus structural variant color for next generation sequencing for selected families from 1000 Genomes project.
Hello, my name is Mateusz Szylinski. I'm a member of Laboratory of Functional and Structural Genomics at Center of New Technologies, University of Warsaw, Poland. Today, my talk will be about the tool we developed, Consensus SV, Consensus Structural Variant Color for next generation sequencing for selected families from 1000 Genomes project. And maybe a little explanation that during our research, we've actually found some tools that did allow the researcher to get the consensus structural variants. However, we didn't really think that they are flexible enough for our research, so we really wanted to actually add that part, and that was the main goal for the tool. And I really think that we've actually achieved that. So we started our research by doing some SV callings. As you can see, we've used a structural variant engine for 5 of the callings: the breakdancer, breaksec, innovator, deli, and lumpy. And the remaining ones were called independently, including Genome Stripe, Manta, Novo Break, Pindel, Svettler, Tardis, and Pump. So after we did all those callings, that's when we actually tried to look for those consensus tools, and then we started developing of the consensus SV. On this slide, you can see what we actually created. As you can see, there are the callings, which are not really part of the whole software, but more like input to that, and you simply need to do it independently. Then we use BCF tools for the pre-processing, and it's pretty much the only requirements, except for the Python, of course, for the tool. Because, as you know, most of those tools produce really, well, of course, they all produce VCF files. However, their structure is sometimes a little different, so we simply extracted the important part for us and went ahead. And then we started looking for SVs that are close to each other, mostly in terms of position, length, and type, because that's what really interests us. And those SVs are called in terms of the next step candidates, and we simply try to establish if majority of them are the same in case of the position and length. Of course, the whole software allows you to set the minimum threshold on how many candidates there should be. We usually use 3 because if a structural variant is found by 3 tools, then we consider it quite high in quality. So going further, if we find that the majority of those candidates, for example, I don't know, 4 of 5. Are exactly the same, then we simply use one of them to create consensus because, well, it simply makes sense in that case. However, if majority of the candidates differ in terms of the position and length, we use our pre-trained neural network model to establish the consensus. Actually, this is the part that I'll elaborate on a little more, because as I mentioned before, we wanted to make this tool as flexible as one might imagine. So to create your own model, you can use any tools you want for the input. You can use even 20 or 30 of them. It's still okay. But you need to add truth VCF file, which will simply be used for calibrating the model. In our case, we calibrated the model on 6 samples from 1000 Genome project, so I would say that it's pretty good. And if you simply want to use the tools that we use or some kind of subset of them, then you can simply use our model. However, as I said, it's only the matter of actually putting the true value and one more parameter, so the tools knows that you want to recalibrate it, and it's done. After we get the list of the structural variants that are created, the consensus structural variants, the genotype of those must be established. However, in our study, we had only 2 types of genotypes that are interesting for us, so we used the simple voting algorithm. We actually thought about creating some more sophisticated algorithm, but it simply didn't really appeal to us, as well. It was pretty. The voting algorithm is completely sufficient for that. Well, the final step is, of course, creating the VCF file with the consensus as is. We also add to the info field the names of the algorithms that were used to establish this one line. I mean, this one structural variant, and they are simply taken from the candidates that were used for the consensus establishing. After creating this pipeline, we actually thought about expanding it further in terms of the expression and functional analysis. However, we thought that it's better to create another tool which will simply be different from the consensus SV, because well, someone simply might not need that, and it's good to keep those things separate in terms of what they do. That's why we created a functional analyzer, which actually takes as an input structural variants, for example, from consensus SV. We also can provide, well, it also can be provided with SNPs and indels. One of those, of course, needs to be put there, because if you don't put any variants, then it's hard to analyze variants. You can put healthy variants, and by healthy I mean common or healthy from another samples, which will be simply removed from the sample in the preprocessing step. This tool for the preprocessing uses BET tools, so they are also the only requirement, like in the previous tool, besides the Python, of course, and some of the basic libraries of it. Also, you can put anchor positions from ChayaPet experiments, and then the tool will also do the 2D analysis. And genes annotation file is also required, but the tool, of course, provides by default the one for the newest reference genome. So after pre-processing the files, we start to look for the overlaps between the structural variants, SNPs, and indels, and the areas of interest. By areas of interest, we mean exons, introns, and promoters. And by promoters, we simply take the areas that are up to 1,000 base pairs upstream from the gene. and this is the case of the 1D analysis, but in case the anchor positions are available, we look for the overlaps between anchors and SV SNPs and indels, and then we take all the areas of interest in the CCD where anchor is compromised and put them on the list. Actually, that tool is still under development, and that is that 2D analysis will be still a little expanded, because the anchor is compromised, it doesn't mean that there are some problems within the CCD. So I'll talk about it a little later, but it still needs to be completed. The output of the whole pipeline is the list of the affected areas, of course, presented in the CSV file. Then we do the callings to the APIs, the ensemble API and UniProt API. From UniProt API, we actually extract Reactome data. However, we simply found UniProt API to be well, easier to use for us, so we stick to that, and it's working really good. So the final output is CSV file with the genes and protein IDs and biological pathways that might be altered. Now a little more about the results of the study. We've actually benchmarked it against the samples from the 1000 Genomes project. As you can see there, we filtered SNPs by the quality and DP, and as you can see the numbers went down a little. We also present there the number of SVs that were discovered in the whole process of getting the consensus by the tool that we developed, and the average coverage of the samples, which is pretty basic. I also mentioned the I also mentioned in the previous slides that you can filter using functional analyzer. You can filter out the healthy, common, and family samples, etc. So we actually there's a list of the filtering criteria that we used in our study. And as I mentioned, we removed the common SNPs from the other samples of the family, because we are only concentrating on the children from each of the families. We also removed common variants from Genome 3.0 database. We filtered out the SNPs that had a minimal allele frequency above 1%, and we also removed common variants from DB SNP database. However, we also use CleanVar for getting the pathogenic variants, and we simply kept them into the study because, well, if there is anything pathogenic, then we would want to know that, because that's the whole point of the analysis. In case of SVs, we use TBVAR, and well, they had to be 70% similar, and we remove the SVs like in the SNPs from well common in the other samples of the family. There I can present you some statistics about the consensus SV itself. We use gold set provided by excellent study by Jason et al. And as you can see, the results are pretty good there. What those percentages mean, we simply take the common structural variants between the sets and divide them either by gold set or by our set. And the 83% is well quite a nice results. Of course, we didn't take the final results of them, because they used different technologies as well. We only compared them in Illumina data that they produced. Also, in case they used a tool which we didn't use in our study, we also filtered it out. And as we used we consider SV to be good, as I mentioned earlier, that 3 tools needed to actually report that. We also filtered out their set that, well, at least 3 tools had to report the variant. So as you can see, it's really good result. We are really happy about it. Well, in case of our set coverage, the difference in numbers might be also caused by the fact that we also use some tools that they didn't use. Well, we'll still need to filter that out and check that out. In case of variants that are unique for the children, You can see the results there, and they are pretty okay. And they were actually put into the further analysis for the functional analyzer pipeline, as you could see there. And the final output actually of the functional analyzer, as I mentioned earlier, is a CSV file. However, we created a simple R script to get some cumulative statistics. And this is an example of the table it can generate, because those are the genes and biological pathways in CCDs affected by structural variants. Of course, we can also generate a table like that for 1D analysis and SNPs and well, all the combinations there. So that's one of the ways to analyze the final CSV file. However, as I mentioned earlier, we still need to do some research on how to decide if SNP is non-synonymous, and also we want to connect the expression of the gene with the actual variants. We thought about using QTLs, but we still need some time to actually polish the tool so it uses all the available all the possible information there for the analysis, and I guess it will be final and published in the upcoming months. So that would be all. Thank you all for your attention, and if you have any questions, I will be happy to answer them.
So thank you very much, Mateusz. If there are any questions, please again raise your hand, and I will call on you to ask. Yeah, and Mateusz is here. Since no one, I'll ask a quick question. There's no hands yet. Are you going to integrate things like proteomics data into your analysis to look at gene expression influence with SNPs, or is it just on the DNA level and RNA level that you're looking?
Well, for now, I think it will be only on the DNA level. However, well, we maybe will develop it further using other data sets as well.
Okay. It doesn't appear that there are any other questions. I'll just give everybody a few more seconds to raise their hand if they have anything. Guess everybody's burned out after the first session.
Yeah, I guess.
It's okay. Okay, great. Thank you everyone for attending the morning session. We will gather again in an hour and about 10 minutes. So that will be 1:20 Eastern Standard Time or 7:20 in Europe. If the organizers have anything else to say, I guess now is the time for that. But thank you.
No, I think we're set. Thanks very much, Michelle.
No problem. It's great. Okay, see you guys all in a bit.